Digital droplet PCR affords enhanced accuracy and sensitivity compared to quantitative PCR. This is a simple method for digital droplet PCR, requiring minimal equipment and no expertise. Particle templated emulsification is a microfluidic-free method of emulsification that does not require any specialized equipment. Additionally, emulsification occurs over a period of seconds and scales to container volume. For commercial beads, add 0.5 grams of dried polyacrylamide particles compatible with particle template emulsification, or PTE, to 30 ml of sterile water in a 50 ml conical tube and mix well. Incubate at room temperature for 30 minutes. For the microfluidic fabrication of beads, pour 1 ml of photoresist onto the center of a 3-inch silicon wafer and then spin it at 500 rpm for 30 seconds, followed by 1250 rpm for 30 seconds. Place the wafer onto a hot plate set to 95 degrees Celsius for 15 minutes to evaporate the solvent. Secure the photomask onto the silicon wafer with a cover glass slide and expose the wafer under a collimated UV LED for 2.5 minutes. Place the wafer on a hot plate set to 95 degrees Celsius for 5 minutes for post-exposure baking.